All right. Chapter 20, Part B. So basically just wrapping up the chapter here, y'all. Not as long as the first lecture, just a couple of pages to cover. So we left off at the end of Part A by basically talking about Darwin's ideas about evolution. And let's try to talk about um, basically how his ideas have been accepted and modified over time. And we'll talk about some of the ways that new discoveries in science and biology specifically have been used to expand some of Darwin's concepts. So we'll begin by talking about Darwin versus Mendel. So you remember Mendel, so the 19th century monk who did some groundbreaking work with breeding pea plants. So he was the first to discover or at least publish about some of the principles of inheritance. So his book wasn't really well received at the time. It wasn't until after he was dead that it caught on. And so in the early 20th century, Mendel's works became more well known. And so this kind of generated an issue because we have Darwin's ideas, which people are using to as an explanation of how animals change, so natural selection, all the stuff we talked about last time. But then Mendel is putting forth some principles of inheritance. So a question that needed to be solved in the early 20th century was how do we combine or how do we satisfy both Mendelian genetics and Darwinian evolution? And so that was part of the effort of a lot of scientists in the early 20th century. So population genetics was a new discipline that arose during that time, basically a synthesis of Mendel's ideas about genetics combined with a lot of new discoveries about genes and DNA and genetic variation, and basically combining those into a new scientific discipline. So population genetics looks at the importance of genetic variation as the raw material of evolution. And so this is a highly mathematical discipline. Basically, when you take genetics, those of you that are biology majors, when you take genetics with Dr. Whitney, probably 2 years from now, you'll really dig into genetics and do a lot of mathematical modeling about how alleles are passed on and about how basically tracking inheritance from generation to generation. And so population genetics focused on this and looked at how natural selection influences uh, inheritance and allele distributions and frequencies. And so this gave rise to the modern synthesis. So the modern synthesis is a unified theory of evolution that integrated data from biogeography, morphology, embryology, paleontology, geology, taxonomy. And basically, what it did was it helped create like a unified evolutionary paradigm under which biology could operate. Because up until this time, geologists were doing their thing, anatomists and morphologists were doing their thing, embryologists were doing their thing, paleontologists were doing their thing, and so on. And so the modern synthesis helped fuse those ideas. And it came with a lot of argumentation and different people promoting, competing theories. But the end result was a more unified synthesis of ideas that embraced a lot of Darwin's ideas, such as gradualism and natural selection, um, and basically, again, just help solidify evolution as like the paradigm under which we do biology. So the modern synthesis also tried to link two different levels or scales of evolutionary change. So those are microevolution and macroevolution. Maybe you've heard these terms before. Microevolution is usually defined as small scale genetic changes in populations, right? So the shift in bill size that we talked about with the finches, that would be an example of microevolution. All right, a cheetah getting faster and faster, that would be microevolution. Dogs or wolves turning into dogs would be microevolution. Well, I guess. Macroevolution is just a larger scale of evolutionary changes. And so in macroevolution, we're looking at new species arising. We're looking at how, maybe whales transitioned from land animals to aquatic animals, or how dinosaurs evolved into birds. So macroevolution is usually above the level of genus or species, whereas microevolution, you're usually looking at changes within genera or within species. And I want to emphasize too that these are not like different things, they're just different phrases to communicate the scale at which you're operating. So it's kind of like Google, what do you call it? Google Earth. So you can be zoomed in at like not population. You can be zoomed in at street level, right? So So in Google Earth, you can like be walking down your neighborhood street, right? So that's like microevolution. You're zoomed all the way in. With macroevolution, you're just zoomed out, and you're looking at microevolution over long, long periods of time. So this would be the equivalent of in Google Images when you zoom out and you're looking at like your region or the state or the country. So not different processes, just different scales of reference. So let's talk about basically how evolution has kind of taken shape as a component of biology since the modern synthesis. And so we're going to hit a couple of the highlights. The first one is adaptation by natural selection. And so under evolution in modern times, basically almost all traits and characteristics of organisms are thought to have been shaped by natural selection. So the example the book gives is the wings of birds, right? So most birds have wings that allow them to fly. I mean, again, there's some that don't, like ostriches or penguins. But regardless, these wings have been shaped by natural selection in ways that allow whatever species of bird we're looking at to survive and reproduce to the best of their ability. So in the case of the ostrich, those wings have been modified by natural selection so that they're not flight wings. And for the penguin, they've been modified so they help the penguin swim. They act like flippers. And in a sparrow, the wings have been shaped by natural selection in a way that allows the sparrow to fly and escape predators and seek out shelter and so on and so forth. The book also talks about how natural selection can sometimes operate on a slow scale. So an example of microevolution would be the resistance that populations of insects have when a new insecticide is applied. And again, you're probably familiar with this concept. You've got insects eating your crop, and so you spray some insecticide on it. Maybe that kills 99% of the population, but that 1% has resistance for some reason. And so with 99% of its competitors gone, that the individuals in that population that have resistance are able to survive and reproduce in great numbers. And over time, your population will change. It will shift from a population that's susceptible to the insecticide to one that is not susceptible. So that's just a small example of how natural selection can operate in the short term. The fossil record is also used to support Darwin's ideas of evolution. So again, Darwin says that all species that have ever lived are genetically related. So if that's true, what we should see in the fossil record is organisms changing within time. And as an example of that, on the lower left-hand corner, I've got a picture. I guess, where did I get this? I think I stole it from Google Images. These are different fossils. Teeth and leg fossils are illustrated along with an artist's interpretation of what the organism looked like. These are all different horses. So these stretch back about 55 million years. And what happened over these 55 million years in the horse lineage is that the horses began as small, little, almost deer-like creatures with teeth that suggested they ate leaves and were more like browsers. They walked around on four fingers. And then over time, they got progressively larger and more adapted to open grasslands. Their teeth shifted so that they were more um, able to efficiently process cellulose-rich grass. Their feet um, changed in a way that allowed them to walk on to change from 4 toes to 3 toes to eventually a single hoof, which is basically their middle finger. And so the fossil record helps trace the evolution of horses over time. The picture on the right is showing plants, but I guess I'll just skip that for now. Another way that we can use fossils, we can use it not just to see how animals change in time, but we can use fossils to help recreate past ecosystems and to look at how the Earth was different in its past. So this is something I put together a few years ago. So on the left is a drawing of this ancient fish that looks a lot like a shark called Helicoprion. And you can see it, again, it looks a lot like a shark, but it's not. So there's a bony fish. It doesn't have a cartilaginous skeleton. And you can tell that its teeth are really weird. It's got these teeth that kind of grow in this helix, hence the name Helicoprion. And just a really pretty cool fish. Anyway, so it was around 300 million years ago, and it's been long extinct. And I was into this fossil, so I just plugged, there's a website where you can plug in a fossil and it shows you where the fossils have been formed, or have been found, I'm sorry. And the map on the right, each of these orange dots represents a place where Helicoprion fossils can be found. So that begs the question, why do we find these, I said shark fossils in the PowerPoint slide, why do we find these fish fossils in the American West, right, which is like the Nevada desert, right? And so the idea is that this used to be an ocean. And so here's a map I got from a different website that catalogs, using different reconstructions, they've tried to put together what North America looked like 300 million years ago. And so it turns out 300 million years ago, there was an inland sea that covered much of North America, right? So much of the Midwest was underwater. And so if we overlay that map with the dots we saw previously, where we find these Helicoprion fossils, you can see that it makes sense that we find these fossils where we do. We find them in the modern desert because the modern desert 300 million years ago was under the ocean, under a shallow sea. Another example, not as many fossils, not as many dots on the map to show, but this is an early reptile, also from 300 million years ago, and we find it not too far from us in Tennessee. We find it in the Kentucky or the West Virginia Appalachians. Right? And so if we take that yellow dot and place it on a map from, oops, no we don't, sorry, we don't do that. And here's another one, sorry, moving on. This is a lepidodendron, which is a seedless vascular plant, also from about 300 million years ago. And from the red dots, you can see where it is. It lived in this stretch, or its fossils can be found in this stretch of modern United States. And then overlaying that on the map, you can see that it liked to live on the edge of this inland sea. And so it lived, kind of in wet, swampy areas. And it would have been buried and fossilized when the sea levels rose, and then it would have grown back when the sea levels dropped, and so on. So and it also turns out that the North America was in a different location 300 million years ago. So this black line represents approximately where the equator was. So you can see that lepidodendron liked to live in hot, humid areas. So not only did it like to live on the edges of this inland sea, but it also grew in a fairly narrow band close to the equator at that time. So there it is laid out, and that's what North America would have looked like. You see Tennessee would have been on the equator at this time. And so when you put them all together, you can see that the fish, the reptile, and the plant fossils all help us paint a picture of what North America looked like 300 million years ago. It helps us track where the sea ended and the land began. Okay, so back to the book. Lots of different disciplines are used to study the ideas that Darwin first put forth. So there's so first up is, why am I talking about this? Biological lineages. Hold up, did I skip this? Yeah, biological lineages. So okay, my bad. Let me go back. So in the fossil record, again, fossils should provide clear evidence of ongoing change. And the change we should see is in biological lineages. Biological lineages are just evolutionary sequences of ancestral organisms and their descendants. Evolutionary sequences of ancestral organisms and their descendants. So, sorry to go back, but this horse sequence, that's an example of a biological lineage. All right. Another example is birds. So there's fossils like Archaeopteryx that are these weird feathered um, dinosaurs slash birds. They kind of have one foot in both categories. And so Archaeopteryx, as you can see in the fossil there in the drawing below, is this weird organism that has characteristics of both reptiles and birds. So it's got wings and feathers, which is very bird-like, but it has conical teeth in its jaw, which is reptile-like. It's got a tail. Now, most birds are going to have a tail, but their tail is made out of feathers. So in other words, there's no actual, they can't move their tail like a reptile, but Archaeopteryx could. So it had an actual tail with feathers on it. And so I'm not going to get into more details right now, but this is an example of an organism that basically serves as the beginning of the biological lineage of modern birds. On the right, you can see where Archaeopteryx fits in. So this is a evolutionary tree. So if you can see my pointer here, here's Archaeopteryx, right? And then, so it shared a common ancestor with living birds. Where does Archaeopteryx and birds fit in with everything else? Like we've talked about the rest of the semester, they're thought to have evolved from basically dinosaurs that walked on two legs. So lots of feathered dinosaurs. We'll talk more about that later in the semester. But basically, we can create a biological lineage of modern birds all the way back to Archaeopteryx and to other feathered dinosaurs, and then through the evolutionary tree of dinosaurs. All right, lots of disciplines that work together in order to view data through the lens of evolution. So historical biogeography is the study of geographical distributions of plants and animals in relation to their evolutionary history. All right, so this is kind of what I was doing with the slides of the lepidodendron and the reptile and the helicoprion fish. So in other words, we can study not just how organisms change through evolution, but also how that's related to where they're living. Right? So some other examples would include, we can look at the species on a continent and how they're related to each other, and then how they're different or similar to other species on other continents. So monkeys in South America, for example, have long tails and broad noses. So they all got that from a South American primate or monkey ancestor, primate ancestor. And whereas the monkeys in Africa and Asia evolved from a different common ancestor, and so they have short tails and narrower noses. All right, and in the picture, if you want to look at how we can use fossils to study historical biogeography, we can use fossils to lend credence to the idea that once in Earth's history, the southern continents were connected as a supercontinent. So we find, for example, fossils that are in the middle of South America, and they're also in southeastern Africa. And so why is that? Well, that's because those two continents used to be connected, and that's why those fossils are continuous across them. All right, we find that we can also do that with India and Antarctica and Australia. Note that India used to be in the southern hemisphere, and Antarctica wasn't always sitting at the bottom of the South Pole. Anyway, so another example of how we can use fossils to calibrate the movement of continents and vice versa. Through comparative morphology, we can analyze the structure of living and extinct organisms and compare them. So basically, we're looking at anatomy between different organisms and trying to understand their evolutionary relationship. One tool we use, or tool we use for these analyses, are homologous traits. And so we've talked about this before, but I'll define it again. Homologous traits are characteristics that are similar in two species because they inherited it from the same common ancestor. So for example, all vertebrates that have 4 limbs evolved or inherited, I should say, those 4 limbs from a single common ancestor. Pigs, dolphins, and bats have the same, even though they have legs and flippers and wings, they all have the same bone setup: 1 bone, 2 bones, hand bones, finger bones. And so those would be homologous because that bone structure is shared by a common ancestor. Homologous traits. Okay, so molecular techniques are another tool that biologists use to study evolutionary biology. And so basically an example of that is that the book talks about is how snakes lost their legs. So as you know, snakes don't have legs. Shocker. But the thought is that they used to. Snakes are reptiles. Other reptiles have 4 legs. And so it's thought that in their past, snakes had 4 legs. And they lost those legs over time, starting with the 4 legs and then the hind legs. So we won't get into the fossil evidence of this right now. Instead, we're going to look at the molecular evidence. And so when scientists look at the anatomy of snakes versus lizards, snakes have no traces of limbs or a neck. Basically, their skull ends and their rib cage begins, right? So a lizard, they don't have that. A lizard, their skull ends, and then they've got a neck, and then they've got a girdle where their forelimbs are connected, and then they've got ribs below that or behind that. And so why is this? What happened? How did snakes lose their legs? Well, before we explore that further, it's interesting to note that some snakes have vestigial hind limbs. So in other words, they don't have limbs, but they have something that, they have some structures that perhaps used to be hind limbs, and now they're used for a different purpose. So in the picture on the right, you can see the mating spurs of a ball python. So these spurs help the male python when it's mating. It's helped the female too. I don't know, but it helps the pythons when they're mating. Those spurs kind of help them grasp on. They can't, grab each other because they don't have arms. And so these mating spurs are thought to be, basically what the hind limbs of the pythons have turned into over time. So they're called vestigial hind limbs because while they're a structure that serves a purpose, helping in mating, they're vestigial. They're not used for the same purpose that they used to be, which at one point in time was for walking around. Oh, and a little note about this guy here. That's it's not a snake actually. It's oh shoot, is it a newt? I should have looked this up. It's a newt. So that little guy, he used to have 4 legs, but he's lost his hind legs, but he's retained freakishly small forelimbs. Just thought I'd throw that in there for you. All right, and so, and one reason I show that too is there's some fossils of snakes that have these really teeny little weird forelimbs and hind limbs, like you can see in this artist's reconstruction of it. But anyway, how did the snakes lose their legs? Well, we can use molecular techniques to try to figure this out. So by looking at genes and the products of these genes produce and the way that these genes shape the development of an organism, we can see that there's 2 Hox genes that come into play when snakes are developing. So these are HOXC6 and HOXC8. And if you look at figure D here, you can see that in this organism, so this is a chicken, it's going to have 4 limbs. All right, and the reason is because HOXC6, the orange one, is being expressed right in that area right behind the 4 limbs, but HOXC8, the blue one, is not. So when orange is on and blue is off, you get forelimbs. That's the point, right? And behind that point, you get ribs. But if you look at snakes, you can see that the blue area, HOX-C8, it is expressed beyond where HOX-C6, the orange one, is expressed. So in that picture on the right, you see the blue extends beyond the orange. So when that happens, the HOX-C8 gene basically cancels out HOX-C6, and the end result is there are no forelimbs produced. So snakes lost their legs, at least their forelimbs, because of a difference in expression of these two genes. So that's kind of interesting because this isn't necessarily, you know, a buildup of information or mutations that led to the loss of the forelimbs. It's a change in the way that already existing genes are expressed. Right? It's also why the hind limbs still persist as the mating spurs in the pythons that we saw, because in snakes, you can see that the same thing doesn't happen in the hind legs that it has in the forelegs. The HOX-C8 gene doesn't suppress the formation of the hind limbs. So the point is, we can use molecular and genetic analysis to understand how organisms change in time. We've also been able to use molecular techniques to potentially do some interesting things. So we've been able to sequence the genomes of extinct species. So the woolly mammoth is a good example of this. So they were able to get enough material. They were able to get genetic material from frozen mammoth remains and sequence the DNA. But the problem was they were only able to get some of the DNA. So I believe it was about 13 million base pairs from a mammoth that was extinct for about 4,000 years. And when they compared that genome of the mammoth to that of an African elephant, they found that the two genomes were 98% identical. All right, and so that's interesting because one, it confirms that woolly mammoths were really close relatives of African elephants, and two, if we're missing places or missing sections in the woolly mammoth DNA, then we can fill it in with African elephant DNA. And just a side note, the picture on the right I stole from Google. This is just showing how one day we might be able to clone a mammoth. So since we have the mammoth genome, what you could do is take that genome and place the mammoth genome into an empty elephant egg. So you take all of the genetic material out of the elephant egg and you replace it with mammoth DNA. And then you take that egg that's got mammoth DNA and you implant it into a female elephant. And basically, you can read through the graphic there, skipping a few steps. But the idea is that eventually you can have an elephant, a modern living elephant that gives birth to a woolly mammoth. So that woolly mammoth's mammoth parents, its biological parents, will have been, long dead and extinct for thousands of years, but it'll be live and kicking. So maybe one day in your lifetime you'll be able to go to a zoo and see a woolly mammoth. I don't know. Who knows? It'd be cool. I'd go. I'd pay money to see that. All right, and we've also used these molecular techniques to study things like the ancestry of dogs. So this is a study that did some sort of molecular analysis. I don't remember if it was DNA or mitochondria or what, but basically they were able to track how dogs were domesticated. So what breeds are most closely related, and basically compare sequences in living dogs to sequences in living wolves, and basically try to establish how we got from wolves to chihuahuas. It's crazy. All right, and then finally, evolution is basically the core of modern biology. So it pops up in almost every subdiscipline of biology in some form or fashion. But it's changed a lot too. So understand that scientists' ideas have changed and will continue to change. And one idea that's changed is that of orthogenesis. Orthogenesis is just progressive, goal-oriented evolution. So this is the idea that evolution is like progressing forwards. It's increasing complexity. It's increasing diversity in a way that's ultimately going to produce some goal at the end. So, and most frequently, that goal is that one day it will produce, humans. So, like humans are the crowning achievement of evolution. But um, most modern biology basically doesn't accept orthogenesis and says that evolution isn't proceeding towards any goal, but it's just this dynamic adjustment to changing conditions and parameters. And also, if you're looking at the pictures of the horse on the screen, note that earlier I showed you the picture of the horse lineage that looks something like the one on the left here that I've drawn a red X through. So just know that scientists don't usually think that the species at the bottom evolved into the next one, which evolved into the next one. So in other words, evolution isn't linear. It tends to be more like a tree. So the picture on the right is a better representation. And so you can see there's lots of splitting, that there may be more or less linear progressions from one species to another, but there's a lot of branching. There can be crossbreeding. So building evolutionary trees and establishing evolutionary lineages is much more complicated than just A leads to B leads to C. Um, so anyway, that wasn't a great ending, but it was an ending. All right, see you next time.